All right, so we need a cladogram so we can talk about, like cladograms. And I wanted to put an accurate one up here, so I'm going to go put some organisms up there, and then we're going to try to identify certain things. Okay. We have fish, sharks and rays, snakes and lizards, birds, amphibians, snakes and lizards, birds, crocodiles, monotremes, marsupials, and placental mammals. Okay, so this is a cladogram. This is not a phylogenetic tree. The difference between cladograms and phylogenetic trees is really technical. A phylogenetic tree will have like spacing for time, and you'll be able to count backwards and see how far back something had branched and something had split. A cladogram just shows you relative differences and relative like relatedness. So this is more of a cladogram than it would be like a phylogenetic tree, but it still works, right? Okay, now we can put characteristics on here. Be careful with characteristics, okay? Because characteristics never happen at a branch point. They happen before or after a branch point. So we could put a little line like right there and say at that point, there was some characteristic that was evolved. This is an ancestral characteristic. Ancestral means that it is that everything past that point has that or has since lost it, and they did have it and then they lost it, and I'll explain what that means in a little bit. What is an ancestral characteristic of fish, sharks, rays, amphibians, snakes, lizards, birds, crocodiles, monotremes, marsupials, and placental mammals? Anybody know one? Something ancestral. Will be a common characteristic of all of them.
Wouldn't it be fins? Like gills or whatever?
I don't have gills.
They all have a brain.
Oh.
A brain, okay, but so do like, well, it's okay, like so do things like worms and crabs and stuff like that. All of these are in I would say subphylum Vertebrata. So these are vertebrates, backbones.
Okay.
And I did have gills when I was about this big, and then they turned into my vocal cords. It's where our vocal cords come from. They come from our ancestral gills, but that's okay. All right, so then we get things like derived characteristics right here. And what would be a characteristic that we could put right there that fish, sharks, and rays don't get, but everything else does? Amphibians, snakes, lizards, birds, crocodiles, monotremes, marsupials, and placental mammals.
They can't breathe on land, right?
So maybe, but some amphibians can, and so that one would work. Like we could certainly do that. I think a more interesting one would be limbs, right? So we have limbs. Or limbs. Okay, as a derived characteristic, that kind of makes sense because all those have limbs except for snakes. So what happened to limbs in the past of snakes at some point in time?
They disappeared.
They disappeared, and that's fine. You can lose traits, like through evolution, traits can be lost. So then we could go up here and say no limbs, and that's all right too. So they limbs were evolved, and then limbs were lost in the branch that goes towards snakes. There's this idea in evolution where like the simplest explanation is probably the correct one, and so there's two options here. We could have evolved limbs a bunch of different times. We could have evolved limbs here, 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 and here, a bunch of different times, or we could have evolved limbs one time and then lost it one time. It's this thing called parsimony, where it means the simplest explanation is probably correct, and it's more simple to evolve limbs once and then lose it to make snakes, and than it is to evolve limbs like 5 or 6 different times. So, but of course, legless lizards aren't snakes. Do y'all know what legless lizards are?
Skinks.
Skinks are close, like they're
Okay.
similar because some skinks are on their way to like legless lizards. Let's see if we can find I found there's a North Texas skink that I found in my front yard one time, like last year.
Ooh.
And it had what you would consider limbs, but its front limbs were useless, basically. Wrong one. Okay, so legless lizards, and you can even tell the difference when you look at like the face of one of these things. Let's see if we can find. The head and the face are very much not a snake face. That's like the face of a lizard right there, and not a snake. But that means leglessness has evolved at least twice. What is that called when similar environments will create conditions to have something evolved twice?
Convergent evolution.
Yeah, and what do you call those structures, or in this case, lack of a structure that has evolved twice again?
Analogous.
Excellent. Analogous structures, or as a result of convergent evolution, instead of homologous structures as a result of divergent evolution. So that's good. That's excellent. All right, let's get back to the cladogram though, because there is stuff on there that we need to add. Okay, if we wanted to put a few other characteristics, we could put things like, what would we put? Do y'all know monotremes, by the way? The groups of mammals, we have 3 groups of mammals up there.
Platypi. Like,
So
are
that
they
would be-
laying eggs?
Yeah, those are the egg-laying mammals. There's only 3 of them. There's the spiny anteater, the echidna, and the platypus, and they were the first branch of animals that had not branched off from laying eggs yet. But if I wanted to put a triangle around mammals, it's not a perfect triangle, but it's just because of the way it was drawn. But there's a name for this. It is called a clade. It includes an ancestor and all descendants. The ancestor is right there, and the descendants are all the ones that branch off of it. There's another word for a clade. Okay, it is called a monophyletic group. And anytime that you have systematicists or taxonomists talking about biology, they try to talk about things in the form of like monophyletic groups and clades. That doesn't always work. Do you guys know what the science of herpetology is? Like, what do herpetologists study?
Amphibians?
And reptiles. I was about to pull it up. So they study amphibians and reptiles, right? Why would we include a science that studies amphibians and reptiles when reptiles are closer related to mammals and much closer related to birds, and yet we have this science that studies them? And that doesn't make any sense at all, right? Like, because they're so different. It just happens they live in, they're small and scaly and they look kind of alike, but and so that means that like a lot of people want to change that, make it some sort of a different science. And there are these other like ways of grouping things that people don't really like. So my cladogram is going to get really messed up here. Okay, which groups are warm-blooded?
The mammals and the birds?
Okay, so if mammals and birds are warm-blooded, how did that happen? Did it happen? Did warm-bloodedness evolve right here? And then was it lost here, 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 right there? Or did warm-bloodedness evolve twice, once here and once here?
Twice?
Twice.
Warm-bloodedness evolved twice. That's that parsimony thing where the simplest explanation is probably correct. So if I put some sort of a shape around warm-bloodedness, it would have to look like that. And there's a name for groups like that. Like if anybody talks about warm-blooded organisms, that is called a polyphyletic group. Okay, a polyphyletic group is the result of convergent evolution. Again. And then we have the problem with the word reptiles, right? If we use the word reptiles, what groups would I include on this?
Snakes and lizards and crocodiles.
But why don't we include birds with that, right? Because shouldn't we? If snakes and lizards are closer related to birds than they are to crocodiles, then we should. And so we all of a sudden have a different type of group with just the term reptiles that would include that. And that's missing a branch. It includes a common ancestor, but not all of the branches from that common ancestor. And that is called a paraphyletic group. That is a common ancestor and some descendants. I always had trouble remembering this, okay? And I finally realized where it comes from. The paraphyletic group, that prefix para, is the same thing you would see in the Paralympics, or the Olympics that happens with people that are like missing limbs or something like that. And para means like, it means, I don't know if it means half, but it means like some of, but not all, or, you know, just different basically. And so a paraphyletic group is that same root word, and the word reptiles is a paraphyletic group. Should we change the definition of reptiles to include birds? Remember, we make these definitions. Like, it's not like anybody can, you know, tell us not to. Like, there's a whole group of scientists that just redefine these things all the time. That's their job, right? They reclassify things. And we kind of have. Like, it turns out we are actually, now, if a taxonomist is talking about reptiles, they are including birds here. By the way, where would we put dinosaurs on this?
At the bottom on the main line.
Well, but what are they closest related to up here?
Oh. Birds?
Yeah, birds. And so that's where you get to the problem of this being a cladogram and not a phylogenetic tree. So we could add dinosaurs, but they would have to be added, trying to pick a different color, right there. Okay, so that would be dinosaurs. We can show that it doesn't make it all the way to today, but then that would also show us like how similar birds and dinosaurs are, and why birds are technically dinosaurs, which is a subcategory of reptiles. They just happen to have larger scales that we call feathers, and they're known for flying. So, all right, what's next? Okay, so let's say we wanted to like use this to maybe build a cladogram or like figure something out, right? So this is a simpler cladogram, and I'm going to just abbreviate these things. Well, no, you won't know what they are. Gorilla, gorilla, hand, meniscus, hand, troglodytes and Homo sapiens. Okay. So this is some of the great apes. I didn't put orangutans on here, but you guys would, I'm assuming, know what gorilla is. Anybody know what pan troglodytes or pan paniscus is? As our closest relative.
I think troglodytes is chimps.
It is, and then pan paniscus is the bonobos that look just like them. And we always say, like it's a really common thing to say, oh, the human's closest relative is a chimpanzee. It turns out that's not really true because we have, nope. That's the wrong one. It's really difficult with 2 keyboards and 2 mice and everything. Let's see. Chimpanzee versus bonobo. There's apparently some size difference, but there's no way that like you or I would be able to distinguish chimpanzees from bonobos, and they look just like them. They're really similar, but we would have no idea of telling them apart. And so when we talk about chimpanzees being our closest relative, that's not true. What's our closest relative?
Bonobos, right?
And chimpanzees, right? Because both of them split at the same time from us, but are we chimpanzees' closest relative?
Yes, right.
No.
No. There's all the other monkeys. I'm sorry.
No, none of these are monkeys. We don't have tails.
Okay.
So we are equal. Okay, let's put the math up here and then figure out how we would have known this. Because think of it like if you have a cousin and your cousin has a sibling. You're equally related to both of them, but they're closer related to each other than they are to you. And that's exactly what bonobos and chimpanzees are. They are the siblings, and we are their cousin. Okay, so the evidence that we would use to make this is DNA evidence. This. Well, we would have lots of evidence. We would have fossil evidence. We would have let's see, what else would we have? Homologies. Homologies and let's see, molecular data. And that's what we're talking about here with a molecular clock. By molecular clock, I mean you can use changes to molecules over time to date things, because our current understanding of DNA replication is that the mutation rates haven't changed over time. And if your mutation rates are constant, then we can put mutations on here. One here, one here, one here, one here, one here. Okay? So each one of those represents a mutation or a DNA replication that happened differently in that branch than another branch. So if we go back from Homo sapiens to the common ancestor of Homo sapiens and chimpanzees, how many mutations different are there? To go back from here to there, how many mutations? We go through 2 lines, right? 2 lines representing 2 different DNA. Now, this is not like accurate to numbers, but there's 2. But what if we go back to the common ancestor of humans and gorillas? How many? So that was 2, and that's 3. That's how we build these. We take DNA evidence along with everything else, and we know that if there's more mutations, then it's been more time, so that branched further back in time. But if we wanted to compare chimpanzees to bonobos, which are pan paniscus and pan troglodytes, how many mutations different are they? Somebody's got to help. To go from here to here.
Only one mutation is
Only one.
Yeah.
So we've only got 1 right there. And that gives us all these relative, like levels of of relatedness, so we can build these kind of things. So we can build it with structural evidence, DNA evidence, protein sequence evidence, and that's what we're going to do with our assignment. So we're going to shift off of this, and we're going to go over to our assignment. Leave the bonobos behind. Right here. So I tried to make this, like a few of them I help you with, and I don't know if we're going to finish this, and I don't necessarily want it to be like homework or anything like that. I want to just put you guys in breakout groups and see how that works. So I'm going to open up mine, and we'll take a look at it, and then I'll put y'all in groups and say go get to work. So you are going to make cladograms. That's the goal here. Let me present so that it's a little bit bigger. Okay, let me show you the slides first because I don't know that we'll get through everything, but the first one, right, where is it? Oh, I don't know why it's changing on that screen, but not on this screen. Am I losing my connection? Okay, I'm gonna do something different here, hang on. Stop sharing. Can y'all hear me okay? Like it's stuck on mine. You can, okay. I don't know why it is. How do I make it stop? All right, let's see. That one left the meeting, so I'm still here. Good, now this should work. Present now. My entire screen. Share. Okay, I gotta remember that my entire screen is presented and I can't open up my grade book and stuff like that. All right. Looks like y'all can see. Can you see my screen now? Making cladograms. Okay. So the first one, I gave you a table of shark, bullfrog, kangaroo, and human, and I gave you characteristics where the X represents that they have that characteristic. So your job on this one would be to type in the names of the organisms at the top, and then the characteristic on the side or on the branches, on the ancestral characteristic and derived characteristic. So that's your first one, because we can make cladograms from tables of morphological characteristics, but we can also, for the second one, make cladograms from genetic information, such as gene sequences. Okay, so this time you have to count up the number of mutations, and so you're going to take the ancestral cell, and that's going to be back in time, and then you're going to count up the number of differences that these have between the ancestral cells. So you have to work together in groups to make that one happen. You're also going to have to make that one on your own right here. I want to see if y'all can use the line like this and the line like this to build your own, because I didn't want to give you a template. Then we have this one. If differences in DNA sequences can be used to make a cladogram, then so can similarities in protein sequence. So these are percent similarity in the protein sequence for a protein called ubiquitin that has to do with DNA, and it's like in everything, which is why it has that name. If we get to it, I've got a bigger table right here, and then I have a table with no characteristics right there. I don't know that we're going to get to that, so maybe we should just target 3 of them, okay? So I'm going to put you all in breakout rooms, just random breakout rooms, open up the assignment, start with the first one. I'll move in and out of the rooms and see if you guys can do this. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So if I go with 3 breakout rooms, you'll get 3 or 4 in each one. No, it's just me.